Hello everyone, this is Dr. P. Mary Anupama, Head of the Department and Assistant Professor of Department of Biochemistry, St. Joseph's College for Women, here with the topic Synthesis of ATP. Objectives of the lesson. So towards the end of the video, the learners will be able to define what is P-to ratio. They will understand about the sites of oxidative phosphorylation. They'll learn about the energetics of oxidative phosphorylation. They'll imbibe the concepts behind hypotheses for oxidative phosphorylation, narrate about ATP synthesis, illustrate the experiments of Paul Bayer's model for ATP synthesis, then the experiments done by Rackets and Masasuke, Yoshida and Kazuhiko Kinosita Junior experiment. The contents of this lesson are, we'll start with the introduction, then P-to ratio, the sites of oxidative phosphorylation, energetics of oxidative phosphorylation, then hypothesis for oxidative phosphorylation. Coming to introduction, oxidative phosphorylation, it is nothing but the transfer of electrons through the electron transport chain to release energy. And here, what is oxidative phosphorylation is? It is the process of synthesis of ATP from ADP and inorganic phosphate. This we call it as oxidative phosphorylation. And the complex 5 which is present in the inner mitochondrial membrane, that is the exact site for ATP synthesis to take place. What is P-to ratio? It refers to the number of inorganic molecules utilized for ATP synthesis for every atom of oxygen which is consumed. So it represents the number of molecules of ATP that are synthesized per pair of electrons transferred through the electron transport chain. Once again, P-to ratio, it represents the number of molecules of ATP that are synthesized per pair of electrons that are transferred through the electron transport chain. So in the mitochondrial oxidation, if at all you get a P-to ratio of 3, then how do you represent it is NADH plus H plus half molecule of oxygen plus 3 ADP plus pi. It will result in 3 ATP molecules synthesis. This is how we represent the equation. 3 means it is indicating that 3 ATP are generated. Okay, and the same way, if it is 2, we assign 2 to FADH2 and not to NADH plus H plus. If it is P-to ratio is 2, then it indicates that it is FADH2. If it is 3, it indicates that it is NADH plus H plus. Now let us see the sites of oxidative phosphorylation. In the electron transport chain, you have 3 reactions that are exergonic and that would result in the synthesis of ATP. So first one, you have the oxidation of FMNH2 by the coenzyme Q. This is the first site which contributes to the synthesis of ATP. The next site that contributes is the cytochrome B and cytochrome C1. Oxidation of cytochrome B and C1, that would result in proton pumping, which in turn contributes to the synthesis of ATP. And the third one would be the cytochrome oxidase reaction. Here also you see that proton pumping is taking place. As a result, ATP synthesis would be taking place. So these are the 3 reactions which are exergonic and that would result in the synthesis of ATP molecules. Now let us see the energetics of oxidative phosphorylation. So as the electrons are being transported from NAD plus or NADH to its redox pair final acceptor, which is half molecule oxygen and water. So here you see that you represent it as half O2 plus NADH plus H plus would result in H2O plus NAD plus. So this is one redox pair and this is the other redox pair. Okay. So the hydrogen is transferred here and finally resulting in water. Okay, and NAD plus is formed. The redox potential difference between these two redox pairs, it is around 1.14 volts, which is equal to 52 calories per mole of energy. And as such, each ATP, it has an energy of 7.3 calories. So if 3 ATP are synthesized during the oxidation of a single molecule of NADH plus H plus through the 3 sites, it is 21.9 calories. And here energy conservation has to be calculated. So what they do is 2.19 into 1000 by 52. That would give 42%. So this is the energy that is trapped in the 3 ATP molecules. Okay, this is the energy that is trapped. Now let us move on to the hypothesis for oxidative phosphorylation. The two most important ones that are proposed are the chemical coupling one and then chemi-osmotic mechanisms. The chemical coupling hypothesis, this was proposed by Edward Slater in the year 1953. And according to this theory, a series of phosphorylated high energy intermediates, these are produced first and they are used for the synthesis of ATP. And these intermediates, these were believed to be like substrate level phosphorylation that occurs in the glycolysis or citric acid cycle that is contributing to ATP synthesis. That is what is chemical coupling hypothesis. Now we shall see the chemi-osmotic hypothesis, which is proposed by Peter Michel in the year 1961. This is the most accepted one and widely understood one. So here the concept is that they compare the energy which is stored here like a battery, wherein you have a positive and a negative charges, and here you have the entire energy which is stored in the battery. So in the same way, here you can see that you have the inner mitochondrial membrane that has the electron transport chain, and during this process we have seen that lots of protons are pumped. But as such, this membrane, okay, it has a high proton density over here, but this is impermeable to the protons. And these protons, they have to be returned to the matrix, or else what you see is that here you have a high proton gradient, while here you have less of protons. And this proton pumping, it has to take place. This proton gradient has to be eliminated, and this proton pumping has to be taken place. So this proton gradient which is generated, that is used for the synthesis of ATP. Here is one more diagrammatic representation of chemi-osmotic hypothesis. So this is the outer membrane and this is the inner membrane. This is the intermembrane space. While this one is the ATP synthesis. These are the complexes, complex 1, complex 3 and complex 4 of the electron transport chain, while complex 5 is the FOF1 ATP synthase. And this is the matrix. So what's happening is, as the protons are pumped through these complexes, a proton gradient is generated. This electrochemical gradient, wherein you have high proton density over here, this cannot cross this membrane directly, but they are pumped through the ATP synthases. So when these are pumped, phosphorylation of ADP is taking place, resulting in the synthesis of ATP. So the energy that is there, the electrochemical gradient which is there, the energy of that is sufficient to phosphorylate the ADP and result in the generation of ATP. That is what is chemiosmotic hypothesis about. The key enzyme that is involved in the synthesis of ATP, we call it as the ATP synthase, and this is our ATP synthase. This ATP synthase is located in the inner mitochondrial membrane. This is the component which is attached to the inner mitochondrial membrane, while here this is the matrix part. It consists of two regions. One is the F0 region, while the other one is F1 region. The F0 one, it actually causes rotation of the F1, and it consists of a C-ring which will be seen. While this F1 component is the one which is responsible for the synthesis of ATP. Here ADP is phosphorylated to ATP because of the F1 component. The F0 part or the F0 component, which is exactly the transmembrane component, it forms like a ring. It is like a transmembrane ring. This F0, it acts like a proton pore that is embedded in the mitochondrial membrane. And this in turn consists of 3 subunits. Here you can clearly see it has 6 C subunits which make up the ring. Then it has subunit B which makes up the stalk that connects to the F1 component, and it also has an A component. These are the subunits. Apart from that, these also have many additional subunits like D, E, F, G, F6 and so on and so forth. So the F0 form, this is the component which is essentially involved in the transport of protons through this one. It forms the pore. Next comes the F1 component. So this F1 portion, it is the mainly important one which is synthesizing ATP. Okay, and it is responsible for the hydrolysis of ATP also. This F1 unit, it will be protruding into the matrix. This region is the matrix part. It has subunits alpha and beta, which makes up the hexamer with 6 binding sites, and 3 of these are catalytically inactive and they bind to ADP. There are 3 other subunits which catalyze ATP synthesis. The F1 subunit also has the gamma 1, the delta subunit and even the epsilon subunit. These are a part of the rotational motor mechanisms. So the gamma subunit, it allows the beta to go through the conformational changes. Okay, that allows ATP to bind and get released. So the F1 is a very large particle and it can be seen through the transmission electron microscope also. It accounts to around 9 nanometers in diameter. That's why you can see them as studs which are protruding into the matrix. So this is the detailed structure of ATP synthases, having an F1 comprising of ABC components, F0 comprising of ABC components and F1 comprising of alpha, beta, gamma, delta and epsilon. Beta are very important. They undergo conformational changes. They allow the ATP to be binding and getting released. Okay, that is how these are very, very important. Paul Boyer is the person who has studied this structure in detail and how it is working in the year 1964. and he identified that conformational changes are taking place in the mitochondrial membrane proteins. They are responsible for the releasing and the synthesis of ATP and all. So the enzyme ATP synthase, it has this F0 F1 complex. We have seen that the C component, it forms the channel, while F1 is the ATP synthesis. So here you have alpha and beta subunits. Alpha 3 will be there, beta 3 will be there. We have seen that. And beta 3 subunit is the one which is holding the ATP or releasing the ATP and so on and so forth, that we have seen. So here this component, it will be undergoing these conformational changes. So the three beta subunits, they exist in three conformations. First is the O, which is the open conformation. Then you have the T conformation and the L. L means it's a loose conformation, while T means it is a tight conformation. Okay, once again, open, loose and tight conformations. So due to the proton gradient, the beta subunit will be in the L conformation. That means loose conformation. That will be allowing ADP and inorganic phosphate to bind to it. Then the L changes to T. The L changes to T. So in T, what happens is ATP synthesis will be taking place. Once ATP is synthesized, then it has to open up and release the ATP. So this is how, what you see is that during the cycle of conformation, it will be the three beta subunits, they will be going through all the three forms. At one time one will be in O, at the other time one will be in T, and then in L conformation. So it is like L conformation wherein ADP and T phosphate will bind, then the T conformation where ATP is held tight, then the open conformation allowing ATP to be released. So this is how ATP will be synthesized. So for one cycle of conformation changes, 3 ATP will be synthesized. For one conformation change of the beta subunit, 3 ATP will be synthesized. So this is what is explained by Pale Boyer in the year 1964. So this is how ATP synthesis will be taking place. As such, when you see, they will be like motors. They will be continuously rotating and rotating. And for every rotation, you see that 3 ATP will be synthesized. So it will be like impellers, the studs will be, or they will be like fans, continuously rotating, and then the protons will be pumped into the matrix and parallelly ATP will be synthesized. That is how these work. This is an interesting experiment which was conducted by E. Rackers. So E. Rackers, he had, and this team, they started isolating fragments of mitochondria. They must have fractionated the cell and they must have isolated or centrifuged, and then they must have isolated these fragments of mitochondria from the cell. And then they started working on it. They could see that these fragments, they were capable of respiration, but they were unable to synthesize ATP. And they also observed that when you add the supernatant of the fractionation, see, they must have taken the pellet which had mitochondrial fragments, but when they have added the supernatant, it had restored the ability of this mitochondrial fragments to perform oxidative phosphorylation. Initially, they were unable to do it, only the mitochondrial fragment sample, but upon addition of supernatant, yes, it is able to do oxidative phosphorylation. So this must have been because they had added something extra to it that led to the synthesis of ATP. The ATP function is restored here, and this part of, or this component of mitochondria which must have got added, that they named it as F1 or factor 1, as it is like a fragment or a coupling factor that is required for ATPase activity. So this F1 factor must have bound to something on the mitochondrial membrane. So it has bound above some factor, so that they termed it as F0, and it was discovered by Yusawa Kazawa. And why it is named as FO or F0 is because it is found to be sensitive to the antibiotic oligomycin. So once both this FO, F1 they were identified, then Racker he was able to confirm that Michel's hypothesis. Peter D. Michels, he hypothesized and he stated that ATP synthesis is not directly related to risk, but it is happening through a high energy intermediate. Instead, it is by a transmembrane proton gradient. So, Racker's experiment has supported or confirmed this hypothesis. So, mitochondrial fragment plus the F0, it is only contributing to respiration. While when you have F1 along with it, which is coming from the supernatant, ATP synthesis is also taking place. Next important contribution is the experiment which is done by Masasuke Yoshida and Kazuhiko Kinosita Jr. The names. So they conducted an experiment. They took a slide and to this they attached the F1 molecule. So FOF1 molecule. And they started experimenting. What they did is to the gamma subunit they tagged it with fluorescent actin protein, and this is to prove that ATP synthesis is taking place as something is moving down. So they started taking pictures at regular intervals, and when this is done they found that it is rotating and then it is contributing to ATP synthesis also. And mainly it is the gamma subunit which is tagged, and they observed that it is the gamma subunit which is rotating. So their observations go like this. Like under microscope they started observing how the fluorescent tag is moving. Okay, and they observed that it is the gamma subunit which is moving with respect to alpha 3 beta 3. So when they immobilized it, it is the gamma subunit which is moving. In the later on studies, F0F1 complex was also used and similar experiments were conducted. And they found that it is not just gamma, it is even the entire C subunit that also moved along with the gamma subunit. And the shaft, which you know, the shaft, that shaft also was rotating. It was rotating 360 degrees. But the rotation was in a pattern. Like at a jerk, it is 120 degrees at a jerk. So this 120 degrees jerk, it was happening at regular intervals of 120 milliseconds. So for every 120 millisecond, say if this is the circle, from here it is moving like this, then like this, and again back. So it is rotating like this at regular intervals. So for every 120, it is moving like this, then like this, and again back. So this is how the gamma subunit is rotating. This was observed as they have taken photographs, and the fluorescent actin filament movement, which is taken in a series of photographs, they summed it up, and finally they have seen that it is the gamma subunit which is contributing to it, and even the entire C subunit is also moving. This is the diagrammatic representation of the experiment conducted by Masasuke et al. So this is a slide, and the FOF1 is immobilized. The actin filament is attached to the gamma subunit. So as it is rotating, the entire shaft was rotating, and parallelly ATP synthesis was taking place. So it was at a jerk. As I've told you, it was at a jerk of 120 degrees at a time. So as it is moving, the entire gamma subunit was moving and the shaft was moving. So here this is moving and this one C is also moving. So thereby contributing to pumping of protons and ATP synthesis. That is how it is taking place. These are the references that are used in the preparation of material. We have Leninger's Principles of Biochemistry by U. Satyanarayana, and I also used Harper's Illustrated Biochemistry to prepare the material and the video. Let us summarize what we have learned. We have studied about P:2O ratio, about the sites of oxidative phosphorylation. We have read about the energetics of oxidative phosphorylation also. Then the hypothesis behind this oxidative phosphorylation. What is ATP synthase? Then Paul Bayer's model for ATP synthesis. Then Racket's experiment, followed by Yoshida Kazuhiko's experiment. So thank you all for watching this video. Please read the material and try to attempt the quiz. Thank you.